We can find them everywhere, in the water, in the soil, and in the air, even in the remotest areas of our world. They are synthetic chemicals that normally do not occur in the environment, produced and put into circulation by industry and agriculture, used by us each and every day, for example in clothing and children's toys, spread around by vehicle exhaust gases and pharmaceutical residues in wastewater. Despite legal requirements, no suitable recording or appropriate assessment of these artificial chemicals in the environment takes place, even despite alarming findings such as the death of large numbers of bees, to which pesticides are likely to have contributed. Researchers at the Helmholtz Center for Environmental Research, UFZ, have made it their objective to identify these synthetic chemicals and to examine their impact on the environment and humans. To this end, they have interlinked their different disciplinary fields of expertise within the scope of the Exposome project. There are over 140 million known artificial chemicals worldwide. Only 22,000 of these have been registered under the European System for the Authorization of Chemicals, REACH, since 2008. An estimated 70,000 substances are in use on a daily basis. These include toxins such as pesticides and herbicides, as well as substances like plasticizers known as phthalates that are used to improve the haptics of everyday objects. In order to understand the effects of chemicals on animals and humans, it is necessary not only to record external exposure, but also internal exposure, which means the substances that are actually absorbed by the body. To identify how chemicals influence the development of diseases, the project's researchers are collecting data as part of the LINA epidemiological study. In particular, they are examining changes at the level of the immune system, allergy development, as well as obesity and metabolic disorders in children caused by environmental chemicals. For this purpose, blood and urine samples from the child participants and their mothers are analyzed in the UFZ's laboratories.
The special feature of the LINA study is that we also included pregnant mothers. This means that we can see at a very early stage which chemicals the unborn child has been exposed to and what implications this has for the child's later disease development. In this way, we demonstrated that children whose mothers were exposed during pregnancy to a higher concentration of plasticizers, known as phthalates, are less protected from allergies.
To establish the environmental chemicals which humans and nature come into contact with, a large number of substances have already been identified on site in the environment, using, for example, large volume samplers, as is the case with the Solutions project on the River Danube. The flora and fauna living there were also included in the study. State-of-the-art analytics performed at the Helmholtz Center's research laboratories then enabled the parallel recording of hundreds of chemical substances in the shortest of times. Tests with algae, water fleas, and fish embryos make it possible to determine the effect of chemicals on a complex organism. In this respect, an understanding of the links between external and internal concentrations is of central importance for our work, as not all chemicals that we find in the environment are also absorbed by organisms. In this context, we supplement chemical analytics with biotests on organisms and cellular test systems that are able to determine the effect of complex chemical mixtures. To be able to cope with the sheer number of environmental tests and analysis of individual substances, the analysis of the environmental samples is predominantly performed using computer-based cellular and organismal tests, applying high-throughput screening methods. For example, robots assume the exposure of genetically modified human cell lines that react to environmental toxins. Within our Exposome project, we are constantly developing new and innovative analysis methods for recording the chemicals present in the environment and humans. Deploying cellular test systems in particular enables us to identify the hazard potential of substances and compounds and thus form a bridge between chemical pollution and the related biological effect. The staff involved in the project have successfully used the findings of their research to pass on recommendations for action to industry, politicians and public authorities such as the Umweltbundesamt. The new findings ultimately make it possible to identify previously unknown chemical compounds and to specify requirements to be placed on industry. Such requirements include, for example, how and in what way a chemical must be structured so that it can be used without endangering the environment or humans. In our Exposome project, we are combining expertise from various scientific disciplines in a unique way. This enables us to perform a comprehensive analysis of chemical effects for the purpose of a preventive risk assessment. After all, we want to continue using chemicals in the future while maintaining the health of the environment and humans alike.